Hello, everyone, and welcome to BPI Digital Week, brought to you by the producers of the face-to-face Bioprocessing International Conferences, visiting Tokyo, Santa Clara, Vienna, and Boston next year. My name is Lauren Berry, and I'll be your host for today's session titled The Multi-Mode Mimetic Ligand Library, a new tool for rapid development of downstream processes. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session. You can also download the presentation slides in the resource list box to the right of your screen. Let's now begin by introducing our speaker, Dr. Steve Burton, CEO of Prometic Bioseparations. Thank you for joining us today, Steve. Now I'll hand it over to you to begin the presentation.
Thank you, Lauren, and hello, everybody. Today, I would like to present a new tool, the Multi-Mode Mimetic Ligand Library, which enables rapid and inexpensive identification of chromatography adsorbents for use in downstream process development. Before we go into the presentation, I'd first of all like to say a few words about Prometic. Some of you may not be so familiar with our company. So Prometic is a Canadian corporation with its headquarters in Montreal in Canada. And the company has a focus on plasma protein therapeutics and small molecule therapeutics. And it also has two operating subsidiaries, which are sort of semi-autonomous subsidiaries, one of which is focused on plasma collection for use in our plasma fractionation programs, and the other is Prometic Bioseparations, which is focused on the development and supply of products and services for use in biomanufacturing. So today we'll be focusing on some of the work that we've been engaged in as a part of the Prometic Bioseparations group. So Prometic Bioseparations, or PBL, is UK-based. We have our research and development facilities based in Cambridge in the UK, and we have our GMP standard manufacturing facilities located just off the north coast of the UK on the Isle of Man. We have been developing and producing chromatography adsorbents now for more than 30 years, and we focus on the high-performance chromatography adsorbents, packed columns, and the development of efficient and cost-effective downstream processes. Currently, in Prometic as a whole, we have around 500 employees, and we have around 100 employees in the UK as a part of the Bioseparations group. So we produce a number of products ranging from small screening column blocks, and I'll be saying much more about that over the next few minutes. We also supply smaller amounts of materials, either in packed columns or in slurry packs for use in process development. We, of course, supply large amounts in bulk quantities for use in large-scale bioprocessing. And recently, we've launched our own range of pre-packed GMP-ready chromatography columns called Evolve-D for use in continuous processing, continuous chromatography. The company has adopted a GMP approach to adsorbent manufacture. We have very high standard production facilities, which are, all of our production areas are ISO 8 standard or better. We are ISO 9001 registered, and we work in conformance with the ICH-Q7 guidelines for the manufacture of active pharmaceutical ingredients. So that's a very high working standard for the production of chromatography materials, and that reflects our commitment to provide high-quality materials for use in bioprocessing. Currently, we have the ability to produce batches of adsorbent from 1 litre all the way up to 1,000 litres. In the photo here, you can see one of our new 2,500 litre reactors that's been installed at the Isle of Man plant, and that gives us a manufacturing capacity for chromatography adsorbent in excess of 35,000 litres per annum. So we're very well placed to be able to serve the needs of the industry. And currently, over 18 of our products are now used in relation to regulatory approved products and processes, both FDA approved and EMA approved. So we're very familiar with the quality standards required to these materials and their applications in biomanufacturing. We have some products that are used now in chromatography columns up to 2 meters in diameter. So again, very well experienced in the use of these in production of biopharmaceuticals. I mentioned just now the Evolve D columns. This is a new product range for Prometic, and these columns are pre-packed GMP ready to use columns, so they enable a plug-and-play approach to column chromatography now. They come fully qualified and they're very simple to use, and at the end of their life they can be then discarded and replaced. So that represents significant time and cost savings to users, particularly for companies that use large numbers of columns or use relatively small columns that are replaced relatively frequently. Those columns are available in a range of sizes. Currently we have column diameters of 7, 10 and 20 centimeters, and we are in the process of developing a 25 centimeter diameter column at the moment. So if you'd like to know more information on those, then please contact Prometic and we'll be very happy to help you. So now moving on to the main body of the presentation. I'm sure many of you are very familiar with bioprocessing, and you'll be aware of the various techniques that have been used to produce high purity proteins. These techniques vary in selectivity. So at one end of the scale we have the filtration type techniques, originally gel filtration, but these days it's ultrafiltration or diafiltration that allows molecules to be separated on the basis of their size. We then have, moving to a slightly higher level of selectivity, we have techniques such as ion exchange chromatography or hydrophobic interaction chromatography, which enable the separation of proteins on the basis of their net charge or their net hydrophobicity. In more recent times, there's been a move towards the introduction of other sorts of ligands that go by a whole variety of different names, but they're commonly referred to as multi-mode ligands. And these are small, simple ligands that generally combine charge and hydrophobic properties. And these are able to interact and differentiate proteins on the basis of their surface charge and surface hydrophobicity and the relationship, the spatial relationship between those charge and hydrophobic groups. And then finally, we have what currently represents the ultimate in protein purification, which is affinity chromatography, where we have affinity ligands that are able to bind selectively to specific sites on a protein, and that selective separation enables very high levels of purification in a single unit operation. So I'd just like to focus now more on the multi-mode ligands, and the way in which these have been used in downstream processes. So not too long ago, downstream processes were generally represented by a series of steps. Each step provided a progressive increase in purity. So it wasn't uncommon to use, starting with some kind of precipitation technique, then ion exchange, HIC, size exclusion, maybe affinity right at the end of the process, and eventually having a protein with the required purity. But that isn't a very efficient way of producing proteins, because the losses of these types of proteins processes tend to be quite high. So these days we use a slightly different approach, where we use generally an affinity capture step right at the beginning of the process to give us a very high level of purification in one step. And then we follow that up with 1 or 2 polishing steps to remove residual impurities. And those polishing steps can be ion exchange steps or HIC steps, but increasingly these polishing steps are represented by multi-mode ligands. And they're finding a niche now in this role, not so much as a capture step, but certainly as a relatively high resolution way of removing residual contaminants in an efficient way. I could spend a lot of time talking about affinity ligands. Chromatic has been working in this area for 30 years plus, and we have a huge experience. I just wanted to share one slide with you just to show how powerful affinity chromatography can be. You're probably all familiar with the use of protein A for the capture and purification of monoclonal antibodies. Well, this slide just demonstrates that we can use the same affinity capture approach with a different adsorbent for other proteins. So in this case, it's a plasma protein. It's a protein called Plasminogen, and we were able to capture with very high efficiency Plasminogen using a designed affinity ligand that we've developed. And this enabled a 340-fold purification of Plasminogen directly from plasma in one step. And that gave us a purity of well over 90% and a recovery of well over 90%. So you can see there that with the right type of affinity ligand, we can develop very effective affinity capture steps for a whole variety of different proteins, not just monoclonal antibodies. So looking now at thinking more about the polishing steps now and the use of multi-mode ligands. So these are small molecule ligands that can participate in 2 or more types of binding interaction. And usually they're small ligands that combine a charge group, either anion or cation, in close proximity to a hydrophobic group, which is usually some kind of aromatic group. These multi-mode ligands can be used as a way of enhancing the selectivity of ion exchanges, and some of you might be familiar with these sorts of, by adding this hydrophobic group, it enables ion exchanges to be used at higher ionic strengths in the presence of higher concentrations of salt, for example. They can also be used for binding an elution of proteins. But that requires adjustment of the ionic strength by addition or removal of salt, or the adjustment of polarity by either adding or removing polarity-reducing agents such as solvents or detergents, and also pH adjustments. And it's not uncommon with multi-mode ligands to have to spend quite significant amount of time with the sort of design of experiment type approach to fully optimize and identify the working window for binding an elution for these sorts of ligands. And that is required because, generally speaking, there are relatively few multi-mode ligands available. The one shown here is actually the GE Capto MMC ligand that some of you might be familiar with. And certainly to apply that one ligand to the purification of any given protein is going to require fairly extensive optimization of the binding and elution conditions. So we were looking at this situation, and we're aware that we have amongst our ligand libraries many thousands of ligands with very different types of protein binding groups, and many of these can be used in a multi-mode fashion. So that led to the idea that, well, rather than choosing a single ligand and adjusting the conditions to suit that ligand, why not take the protein solution that we have, that we need to purify, and find a ligand, a multi-mode ligand, that is suitable, that is optimal for that particular protein in that particular mixture. So it's a very different way of looking at multi-mode ligand chromatography. So what we have done here now with our multi-mode mimetic ligand libraries is we have a much larger array of ligands. I'll show you in a moment our multi-column library block, and we have 96 different multi-mode ligands in that block. And these ligands are typically attached to agarose beads by the use of spacer arms to make the ligands fully available for protein binding. We use a triazine attachment chemistry that gives very high fidelity, so very good synthesis and purity of the synthesized ligand. We use a solid phase synthesis approach to produce these ligands, and that gives us this chemistry gives us alkali stable ligands. So all of these ligands can be treated with 1 molar sodium hydroxide, and the majority could even be autoclaved if necessary at neutral pH. We use a wide variety of groups, so we're not just restricted to 1 or 2 anionic groups and a hydrophobic group. We can use a whole range of different groups here, so anions, cations, hydrophobic groups, hydrophilic groups, or any combination of these. The libraries themselves are packed into 96 column blocks. You'll see some photos of this in a moment. And these blocks are microcolumns, so each one is 250 microliters. So it's 96 discrete columns in a single block, which is in the standard 8 by 12 array that is commonly used for screening and for assays. They are true packed columns, so they have a top frit and a bottom frit, and they can be run in a chromatographic flow-through mode. So these aren't batch adsorption plates. These are true miniature chromatography columns that can be operated in a true flow-through mode. Screening is very simple. We can do this by running the column blocks under gravity, and the target feed material can really be anything. And we can screen either for ligands for capture or for binding impurities. And it's probably the use of these ligands for polishing applications that's the most interesting right now, because for those applications we simply want to bind the impurities and we don't want to bind the target. So as long as the target protein passes through the column unhindered, then and the ligand binds impurities, then that's a potential candidate for use as a polishing step. So these multi-mode ligands now are a little more selective than the traditional multi-mode ligands, and they sit here in this sort of this array of different purification techniques. So they're not quite as selective as the affinity type ligands, but they're more selective than the traditional multi-mode ligands. In terms of synthesis, I mentioned the triazine chemistry, so we can see that here. We start with agarose beads, which are very hydrophilic, very robust, very stable. And then we attach to those beads a chemical activating group, which is cyanuric chloride, and that gives us this dichlorotriazine functionalized material, as you can see here. And then we can attach to those reactive chlorines different amine groups. And these amine groups, the side chains of those amines, can really be anything. They could be an ion exchanger type compound, they could be hydrophobic, or even other types of groups that we use for our affinity ligand library screening applications. When we react the first amine compound, the reactivity of the remaining chlorine is a lot less than the reactivity of that first chlorine. So that means that we can drive these reactions to completion. So we get a very clean synthesis, the attachment of the first amine. We wash away the unreactive materials, and then we can react the second amine with the remaining chlorine to give us this di-substituted ligand product. The types of R groups that we have with these ligands are really at quite a wide range now. So in terms of ionic groups, we can have sulfonates, carboxylates, phenolates, amino, pyridyl, various types of functionalized amino groups here. And in terms of hydrophobic groups, we can have phenyl groups, we can have alkyl groups, and we can have other types of groups as well, such as cyclohexyl groups. So by combining these two groups of these ionic and hydrophobic side chains, we can produce a library of multi-mode ligands that have quite diverse structures and quite diverse protein binding properties. We've organized these ligands into 6 different zones, and the reason for doing this is to make it easier to understand the chemical structures and chemical properties of the ligands in each zone, and how you might approach the binding and elution of proteins from those zones based upon knowledge of the types of ligands that are in those areas. So in zone 1, we have ligands that are predominantly ion exchange in nature. In zone 6, we have ligands that are predominantly hydrophobic in nature. And then in zones 2, 3, 4, and 5, we have ligands that combine combinations of ionic groups and hydrophobic groups. So in zone 2, we have ligands that combine anion and anionic groups with aliphatic groups. In zone 4, we have ligands that combine anionic groups with aromatic groups. In zone 5, we have ligands that zone 3, we have ligands that combine cationic groups with aliphatic groups, and in zone 5, we have ligands that combine cationic groups with aromatic groups. So we have quite a diverse array of ligand structures and different ligand binding properties. We did some work to convince ourselves that these ligands really did have different binding properties. And the way we did this was to analyze the chemical structures of the ligand using some computational chemistry software, the Schrödinger suite. And this software is very powerful, and it enables modeling of ligand structures, protein structures for that matter, and it also enables the characterization of ligands on the basis of a whole range of different chemical descriptors. So we can look for hydrophobic accessible areas on the ligand, and that's shown there in the top left. Blue means it has a very low score for hydrophobicity. Red means it has a very high score for hydrophobicity. And you can see that within those 96 ligands, we have a very diverse range of ligands in terms of their hydrophobic properties. We can do the same for other chemical descriptors such as hydrophilic accessible surfaces. You can see we get a completely different pattern there for hydrophilic ligands. We can look at ligands that can participate in hydrogen bonding in terms of hydrogen bond donating groups, and ligands that participate in hydrogen bond accepting groups. And again, we see very different patterns. So this is telling us that these ligands are different. So when we challenge them with different proteins, they're not all going to behave the same. Some will bind some proteins and some will bind other proteins. We look at more of these types of descriptors. Pi carbons is a measure of the aromaticity. So as you would expect, the groups that have predominantly aromatic groups are lighting up there in red. We can look at overall polarity, log P. This shows us the ones that are mainly hydrophobic. We can look at the number of rotatable bonds in the ligand. That gives us a measure of flexibility of the ligand. And we can also look at predicted binding to different proteins such as albumin. And if you look here, you can see 3 of these patterns are actually quite similar. Log P, the number of rotatable bonds, and predicted binding to albumin all look fairly similar. And you can see there's one ligand in the C11 position that's lighting up very strongly in all cases. And this is because this particular ligand has very long hydrocarbon chain for the aliphatic group. And if we think about albumin, albumin is a protein that binds long-chain fatty acids, which are long-chain hydrocarbons. So what we see here is that this particular ligand at the C11 position, we're predicting it's a good albumin binder because it's relatively apolar, it has a long aliphatic chain, and that makes it quite flexible as well. So this chemical analysis by using computational chemistry, it can be very powerful to predict how ligands might interact with proteins and what to expect when we do screening. So we'll come back to this albumin prediction in a little while. This is what the multi-mode ligand library looks like. As you can see, it's in a conventional 12 by 8 array with the various zones color-coded there. One of the main things to emphasize about this is it's very quick and easy to use. It's not necessary to use a robotic liquid dispenser. If you have one, you can use it, but it's not necessary. You can just use a multi-channel pipette, as is shown here. The blocks run under gravity, so you don't need to use a plate centrifuge to get flow through the beds here. They will just drain all by themselves under gravity. And if you have plate-based assays already set up for your proteins, then it's possible to do the binding and elution studies very quickly indeed. I mean, we typically do these studies in 2 or 3 hours in our lab. And if you compare that to how long it would normally take to run 96 columns, you can see that's a fantastic saving in time. As I said, these are discrete chromatography columns, so they have a top frit, they have a bottom frit. So once the seals are removed, the plates will then drain quite easily under gravity, and then it's just a question of equilibrating the columns and adding the desired feedstock and just letting that flow through the columns under gravity into a collection plate. And we provide a collection plate with the column blocks as a part of the kit. The starting material should be the actual fraction that you want to purify. So that could be your cell culture supernatant, it could be fermenter broth, or it could be plasma or serum or whatever it is. It could be an existing column fraction you have if it's a polishing step that we're developing. The key thing here is that we start with what we want to purify, not some sort of artificial mixture which contains a purified protein in buffer. That doesn't give, generally speaking, a very good prediction of which ligand or which adsorbent will be useful for purification, because what we want to do, as well as looking at the binding of our target protein, we need to understand how other compounds, other impurities that are present, bind as well. So if we want a polishing step, we want to know that the target protein doesn't bind and the impurities do bind. Conversely, if we want a capture step, we want to know that our target protein does bind and the impurities don't bind. And the best way of determining this is by using the starting feedstock that has both the target protein and the impurities present. So in the first example of the use of the Multi-Mode Mimetic Ligand Library, we're going to look at screening of a monoclonal antibody feedstock. So this is a CHO human IgG1 antibody. It's in clarified cell culture supernatant. So we start by equilibrating all of the columns in a phosphate buffer at pH 7.4. We then load our feedstock onto this, so we're loading 1 mil. The columns are 250 microliters. The wells are 1 mil in volume, so we can load that as 1 mil fraction. If we wanted to, we could load more than that, just simply by, after that first mil has drained through the columns, we can then pipette on another 1 mil fraction. So you can do that as many times as you want to increase the loading volume if that's required. After loading, we then wash the columns. Usually that washing is done with the equilibration buffer. And then finally, we will then elute off the bound proteins. In this case, we're using an acidic buffer for elution. Generally speaking, that's because acidic buffers work quite well for antibody elution off a whole range of different types of adsorbents, particularly protein A. So we used an acidic elution step here. We can use other sorts of elution buffers depending upon the nature of our protein. We have here with these multi-mode ligands, because they provide embodied combinations of charge groups and hydrophobic groups, very often an elution buffer that combines both salt and a polarity reducing agent works quite well, and I'll give you some examples of this later on. But in this particular example, we're eluting with a citrate buffer at pH 3. So if we now add just run our antibody through the block and then wash the block and elute off at pH 3. What you're looking at here now is just a summary of the binding of antibody as seen by analysis of the elution fraction. So this is the presence of our monoclonal in the elution fraction. So to be in the elution fraction, it must have bound to the ligands. And what you can see here is that some of these ligands do bind the antibody quite tightly. And other ligands really don't bind the antibody at all, and we've got some that have partial binding. We've scored these just on a scale of 1 to 5 in terms of their ability to bind, and we've color-coded them, and you'll see a lot of this in the next examples, just to make it easier to understand whether any given ligand is binding or not to the target. If we focus just in one area here, so this is zone 2 of the plate, so this includes ligands that are anionic in character and that have an aliphatic group, so they're anions and aliphatic groups combined together in that zone. And if we take those fractions and run them out on an SDS page gel, we can see here that a number of these ligands are doing a very good job of binding impurities, host cell protein. So if we look at lanes 5, 9, and 13, you can see those ligands are pulling out a lot of protein that isn't product related. This is host cell protein. If we look at other lanes, such as lanes 6 and 10 and 14, you can see those ligands are binding some host cell protein, but also some antibody-related impurities. So there's a band here that at around 45 kilodaltons that is a breakdown product of the heavy chain, and we can see the excess light chain at around 25 kilodaltons. And so some of these ligands are not binding the antibody. The whole antibody is much higher up the gel there at 150 kilodaltons. But some of these ligands are not binding the whole antibody, but they are binding host cell protein and they are binding the antibody-related impurities. So they're quite interesting ligands, and we'll be looking at those in a bit more detail in the next few slides. We can also screen these libraries for host cell protein. So now, in this case, we've taken those elution fractions and we screen them using a HCP assay. It's a HTRF assay that we've used to estimate the concentration of host protein in there. And you can see rows C and D in particular, those ligands are lighting up quite strongly for the binding of host cell protein. We can now compare that HCP binding data with our antibody binding data, and if we just look at these now, so in the top left, this is the slide you've seen previously for the elution fractions from the Multi-Mode Mimetic Ligand Library for the whole antibody. Top right, this is the HCP binding data that we've just seen. Bottom left, this is the binding of that 25 kilodalton antibody-related impurity, and bottom right, this is the binding of the light chain. So this is the concentration of the light chain in the elution fraction. If we compare all of these different sets of data here, if we look now in yellow, these boxes represent ligands that have a high binding of HCP and a low binding of antibody and antibody-related impurities. So if we wanted a polishing step that didn't bind the antibody or any part of the antibody, but did bind the HCP, then those ligands would be particularly good ligands to select and use for HCP removal from that particular antibody. Also, if we now look at the ones I've highlighted in orange, these are ones that also don't bind whole antibody very well. They have maybe a small amount of binding for HCP, but not much. But what they do have a very good binding of is the 45 kilodalton impurity and the light chain. So if we wanted a resin that was going to remove antibody-related impurities, then those ligands highlighted in orange there would be a good choice for that application. If we just look now in a bit more detail at those ligands that we highlighted in yellow and in orange, we can see those here. So in lanes 3, 4, and 5, this is SDS page of those elution fractions, and you can see there the presence of a large amount of host cell protein. Of course, HCP isn't a single protein, it's a wide spectrum of proteins that are related to the host cell organism. So it's the cytosolic proteins typically from those cells. In lanes 6, 7, and 8, these are the orange ligands that are binding predominantly product-related impurities. In lane 6 there, this is actually ligand D9. That's a particularly interesting ligand because, as you can see, not only does it bind product-related impurities quite well, it also binds HCP quite well as well. So that's almost a universal impurity-binding ligand. And in all of those ligands, they have very low binding of the target protein. So the full the full antibody is passing through these columns and retarded. So this is an example of how we can use the multi-mode mimetic ligand to select a ligand and adsorbent for the polishing of antibodies, for the removal of either HCP or product-related impurities or both. Once a ligand has been selected from that screening, it's then we can then just contact Prometic. We can then provide larger amounts of any one of these ligands, either in a pre-packed column format in our Evolve D columns, or we can supply any amount of slurry, and we can provide large volumes of these for process applications as well, and we can provide the full support going forward. But the starting point, the identification of the adsorbent, is what we're using this block for, and it's very quick and very efficient for doing that. And I'd like to move on now to some other examples. We're using now human plasma. This is partly because Prometic does a lot of work with human plasma, and also because it's a very complex feedstock. There are tens of thousands of different proteins in human plasma, and it's a very good source of proteins, and you can really see the differences in selectivity that the ligands have, and how we can use these ligands to pull out and remove different proteins from plasma. So we'll start by just looking at general binding of plasma proteins. So in this example, we're starting with equilibrating at pH 7.5, and we're using a citrate buffer containing calcium chloride, and that's because we don't want the plasma to clot when it passes through the column. It's whole plasma, so we have all of the clotting factors here and fibrinogen. So if we mistreated that material, it would form fibrin and it would block the column. We then load under those conditions. We're loading human source plasma to each column. We're then washing with equilibration buffer, and then we're eluting here with this combination buffer that combines 1 molar sodium chloride with 10% hexane diol. And this is a really useful buffer for eluting protein from these multi-mode ligands that combine both charge and hydrophobic groups. That elution buffer contains salt to neutralize charge groups, and it contains hexane diol, which is a very good it's quite soluble at 10% concentration, but it's good for reducing the polarity of the buffer to neutralize hydrophobic interactions. So on the next slide, we have now the SDS page of the elution fractions from the different columns in zones 2, 3, 4, and 5. And you can see here, just by looking at this, each one of these has a very different banding pattern. So if we look at, for example, lane 10 there, you can see that ligand is binding albumin quite strongly. That's that large dark band. But if you look at rows 11 and 12, for example, those ligands bind very little albumin. So each ligand has its own protein binding selectivity. Each ligand will bind a different spectrum of proteins. It's just a question of screening them to determine which ligand binds the protein or proteins that we're interested in. On the next slide here, we can see, again, these heat maps of binding, where we've been looking at specific plasma proteins. So at the top there, these are ligands that we've identified that bind fibrinogen very well. In the next one, the middle one, these are ligands that bind IVIG, so this is polyclonal IgG. You see a different spectrum of ligands lighting up here to the ones that we found for monoclonal antibodies, so there is a difference here, obviously, between human plasma IgG and the monoclonals. And then at the bottom there, these are ligands for albumin binding. And if you recall, a few minutes ago, I presented some data on the computational chemistry modeling, and there was a prediction there that ligand C11 would be a good binder of albumin. And as you can see here now, this is an actual screen for albumin, and ligand C11 is one of the ones that's lighting up very strongly as a good albumin binder. So the prediction matches exactly what we see in practice with that ligand C11. We can also use these ligands as well as binding individual proteins. We can look at the removal of unwanted impurities. Now, in the case of plasma proteins, impurities are actually human plasma proteins. So some of these are useful proteins that we want to have. But there can be cases where there are proteins in human plasma that we actually want to remove. In particular, where we have activated proteases or activated clotting factors, because these can, in the context now of plasma protein therapeutics, these can, if we have activated proteases or activated clotting factors, that can give rise to either excessive bleeding or unwanted clotting, and both of those are very undesirable for a therapeutic product. So there are situations where we want to specifically remove certain types of plasma protein impurities. So in the next few slides, you'll see how we've used these multi-mode mimetic ligand libraries to do just that. So we'll start with an example of the capture and removal of IgA. So this is immunoglobulin A. There are pharmacopoeia limits for the amount of IgA in IVIG products. Excessive amounts of IgA are undesirable, and it's important that the IgA is removed. So what we've done here, we've taken IVIG, we've spiked IgA into it, and then we've used an IgA ELISA to look at the removal of IgA. So this is now looking at flow-through fractions, and you can see some of these are capturing IgA and some of them are letting IgA through in the flow-through. Some of these ligands here have very low binding of IgA. In the flow-throughs, that means they're capturing IgA exceedingly well. So if we wanted a polishing step for IgA, these will be the ones that we'd be interested in. And we've highlighted 6 ligands here that are able to capture IgA, and all 6 of these have very low binding of IVIG. So more than 95% of the IVIG goes through in the flow-through fraction, and the majority of the IgA is captured and bound and removed. In another example, this is now the removal of kallikrein. So kallikrein is a protease when it's activated, and it can catalyze the conversion of plasminogen to plasmin, which is a very potent protease, and that can lead to bleeding episodes, uncontrolled bleeding, if there's activated kallikrein in plasma products. So again, we've done a similar thing here. We've taken whole plasma, we've applied a kallikrein ELISA assay to the flow-through fractions, and the ones in red are the ones that are removing kallikrein, so very low concentration of kallikrein in the flow-through fraction because it's being bound by the column. And in this example, we can see one ligand here in position G1 that's quite effective in capturing and removing kallikrein, but it doesn't bind it has low binding of other plasma proteins, particularly IVIG. Another example here is factor 11. This is a very real concern amongst plasma fractionators. There have been cases a few years ago of very large-scale product recalls of IVIG because of excessive activated factor 11 in the IVIG. What we've done here, we've taken plasma and we have now a factor 11 ELISA. And like we've done previously, we've passed those through the column blocks and we've looked at the flow-through fractions and identified fractions that are low in factor 11, sorry, because that means we've then captured the factor 11 on those columns. You can see here 3 ligands are very effective at capturing and removing factor 11, but they have low binding of IVIG, which is what we want to see. So we took one of those ligands, the ligand at the G1 position, and we did a slightly larger scale experiment. So this is in a conventional packed column now, a 2.5 mil column. So we loaded our plasma onto that column, washed it through. You can see a very large flow-through peak, as you kind of expect to see. And then we eluted off the bound protein, and you can see that quite sharp elution peak there at the point. And the factor 11 is all contained in that elution peak. There's very little factor 11 in the flow-through fraction. If we now look at our SDS page of these fractions in lanes 5 and 9, this is showing the elution fractions of these columns. So they have bound not just factor 11, they have bound a few other proteins. But if we look at the major plasma proteins like albumin, for example, IVIG, most of those are coming through in the flow-through fractions. So this particular ligand here, this ligand G1, is very effective in removing factor 11 from plasma or from IVIG that may have residual factor 11 present. So to summarize now, what we've seen here is that we can use both affinity chromatography as a capture step and then use these multi-mode mimetic ligands as polishing steps, and we can do that very effectively if we have a library of diverse multi-mode ligands. Prometic has developed just that with the Multi-Mode Mimetic Ligand Library. So this contains a very wide range of ligands that combine both ionic and hydrophobic groups, and this can be rapidly screened to identify ligands for either capturing or polishing steps. So this is really a change of paradigm now for multi-mode ligands. So rather than spending a lot of time and effort in the lab to optimize the feed material so that we have binding of either the target or the impurities, what we now do is simply take the feedstock as it is, run it through the columns, and select the ligand that provides the binding properties that we desire. Finally, I'd just like to acknowledge the fact that a lot of work has gone into the discovery and selection of these ligands and the development of the column block. Some of my co-workers here are listed, so Chloe, Graham, Buzz, and Lucy. So a big thank to them and to the PBL research team. At this point, I'll hand you all back to Lauren for any questions that you may have. Thank you.
Thank you, Steve, for an excellent presentation. We've received a few questions already, but we'll give the rest of you a moment to enter your questions in the Q&A box to the left of the slides. Before we begin the Q&A, I'll run through some brief announcements. First, I'd like to thank Chromatic Bioseparations for sponsoring the event. Next, I'd like to quickly draw your attention to our face-to-face BPI conferences, visiting Tokyo, Santa Clara, Vienna, and Boston next year. At these leading annual bioprocessing events, you can hear from hundreds of expert speakers and network with peers. Also, be sure to check out the resource list to the right of your screen, where you can download the presentation slides from this session and a large collection of useful resources. Now, back to Steve to begin the Q&A. So, Steve, we have a question here. Do you have a ligand or ligands which specifically remove or reduce CHO-HCP in a flow-through mode for MAB or FC fusion protein products?
Yes, I mean, one of the examples I showed there was exactly that. So if we look at, I'm just going to move, select one of the slides here. So if we see this particular slide I showed earlier, if you look at row 6 there, that particular ligand is doing exactly that. So it's removing HCP and it's removing product-related impurities from a CHO antibody. So this is I think it's an IgG1 antibody producing CHO cell line. So that's an example of doing exactly that.
Great. Let's bring in another question. Yep, another question that we have here. If I get hit on the plate, what's the next step?
So the next step would be to contact us and let us know which ligand or ligands you're interested in, and then we can provide either ready packed columns, larger columns to do work with. So we have a range of columns that are in 5 ml volumes or 50 ml volumes that are pre-packed. Or we can supply slurry, so we can supply 25 mils upwards of samples of that particular adsorbent so that you can do your own continuing developmental work. And we can supply large amounts right up to 1,000 liters, 1,000 liter batches if that's required.
Great. All right, another question. Can I get the structure of any of the ligands?
Yes, we do. I mean, we don't publicize these at the beginning. But if anybody, any users of the plate that have identified a ligand that they're particularly interested in and they want to know more about it, we'd be very happy to share the structure of that ligand with you. That will probably require some kind of confidentiality agreement depending on the type of information that you was interested in. But certainly we can provide information on the ligand structures. If you're interested in any specific ligand and you wish to take that forward in your process development work.
That's great. Do you supply suitable generic binding elution conditions?
We do. We have a list of buffers that we recommend for each of the zones on the plate. I highlighted this sort of general buffer that we have that combines salt and hexanediol. That's often a very good starting point. But we have other buffers that we recommend, and that information is supplied with the Multi-Mode Ligand Library kit as to what buffers that you can try for elution for ligands in each of the 6 zones.
What if I find a ligand that binds my target, but not at the capacity I need?
That's a good question. So generally speaking, for impurity removal, for polishing steps, the capacity required isn't very high because, generally speaking, it isn't a large amount of those impurities present, certainly not after an affinity capture step. But in some cases, it can be an issue. So if you see a ligand that is giving a very good purification, but the capacity maybe could be improved, then we can develop another version of that, so we can take that particular adsorbent and we can do some investigations. For example, we can increase the ligand loading on the resin to give an increase in capacity, or we might look at a different support matrix. For example, we could look at a smaller bead to give a higher surface area, or we could look at a base bead that might have larger pores if porosity was limiting the binding. So there are various things that we can do here to increase the binding capacity of these resins after they've been selected.
All right. One last question we have here. How quickly could I get a full-scale batch of any absorbent on the plate?
Okay, well, in terms of larger amounts for continuing investigations, we can provide those very quickly. They're off the shelf. Depending upon the scale of the batch that was required, we can certainly supply liter quantities very quickly, and we can provide 100-liter quantities within probably 10 to 12 weeks of that order being identified to us. So we can scale these things up relatively quickly. If there was any optimization required, then that would take a little time in addition to those time scales. If it was just simply a larger amount of the same material that was in the plate, then we could produce larger amounts of that relatively quickly. If there was a need for supporting information, some people might wish to have, for example, 3 batches produced as a part of a validation program. We can do that. So again, there will be a longer time scale associated with that, but we can certainly provide sort of larger batches and validated batches going forward.
Great. That's all the time that we have for questions today. Thank you, Dr. Burton, for a great session. If anyone submitted a question that wasn't addressed, please keep in mind that the speaker will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. Also, don't forget about all the downloadable resources in the resources box to the right of the slides. Before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Connect 365 Life Sciences, have a great day.